How is neurodegeneration studied in clinical research? Have you ever wondered how scientists study neurodegeneration, especially when it comes to conditions like dementia and memory loss? The process is quite fascinating and involves several methods that work together to uncover the mysteries of these diseases. Researchers aim to understand how these conditions develop, track their progression, and evaluate potential treatments. To start, clinical evaluation plays a crucial role. Researchers conduct detailed assessments of cognitive function, memory, behavior, and neurological status. These evaluations help to identify symptoms and the stages of diseases like Alzheimer's disease, frontotemporal dementia, and Parkinson's disease. Next, biomarkers are essential in this research. Biomarkers are measurable indicators of disease processes. For instance, proteins like beta-amyloid and tau found in cerebrospinal fluid are closely linked to Alzheimer's disease. By tracking changes in these biomarkers over time through repeated sampling, researchers can monitor disease progression and cognitive decline. Blood-based biomarkers are also gaining importance due to their less invasive nature. One example is neurofilament light, a protein released during neuronal injury. This can be measured in blood samples, making it easier to monitor various neurodegenerative disorders. Neuroimaging techniques are another vital aspect of studying neurodegeneration. Imaging studies like magnetic resonance imaging and positron emission tomography, scans, help visualize brain structure and function. For example, positron emission tomography can detect amyloid plaques and tau tangles, which are hallmark features of Alzheimer's disease. These imaging tools complement biomarker data and clinical assessments, providing a comprehensive understanding of neurodegeneration. Moreover, researchers investigate the molecular and genetic factors involved in neurodegeneration. They explore the role of small RNA molecules and genetic mutations that may contribute to disease susceptibility and progression. Emerging research is also exciting, as it includes trials of new therapies. These therapies may involve immunotherapies that target harmful proteins, gene therapies to correct genetic defects, and even stem cell therapies aimed at repairing damaged brain cells. These studies relying on biomarker and imaging data to assess treatment effects and disease modification. Finally, advances in remote and longitudinal monitoring techniques allow researchers to conduct repeated assessments over time. This is crucial for understanding how diseases evolve and for evaluating the effectiveness of interventions. For instance, remote blood collection methods, like fingerprick sampling for neurofilament light, enable more frequent monitoring without requiring in-person visits. In summary, studying neurodegeneration involves a blend of clinical assessments, biomarker analysis in both cerebrospinal fluid and blood, neuroimaging, molecular genetics, and clinical trials of new therapies. These combined methods provide a detailed understanding of how these diseases work, how they progress, and how they respond to treatment. This research is vital for developing better diagnostic tools and therapies, ultimately improving care for those affected by neurodegenerative diseases. Always consult healthcare professionals for personalized medical advice.